Hello, everybody. Welcome to Miss McGuire online lecture. And today we're covering cells, the living units. Okay, so let's go ahead and begin. Cells are important structural and functional unit of any organism. It's actually the smallest unit of life. And importance of cells are described in cell theory. Cell theory states that a cell is a structural and functional unit of life. How well we function, how well the entire organism functions depends on individual and combined activity of all its cells. Structure and function are complementary. Biochemical function of cells are dictated by shape of cell and specific subcellular structures. That means if we know the shape of a cell, right, we can predict its function. They are connected, they are complementary. And continuity of life has cellular basis. So again, cell is a structural and functional unit of life. The function of entire organism depends on activity of its individual cells. Structure and function are complementary, and life continues because cells divide. And cells can arise only from pre-existing cells. We have over 200 different types of human cells. Types differ in size, shape, and subcellular components. These differences lead to differences in functions. All cells have some common structures and some common functions, despite of this variety of different types of cells. Human cells have three basic parts. First, plasma membrane. It's a flexible outer boundary. Second, cytoplasm. Cytoplasm is intracellular fluid containing organelles. And nucleus. Now, nucleus present only in eukaryotic cells, and human cells are eukaryotic. Nucleus contains DNA, and it's a control center. Also, we have substances found outside the cells. We call them extracellular material. Extracellular materials include extracellular fluids, such as body fluids, right? Such as interstitial fluid or tissue fluid, that's another name. Cells are submerged or bathed in this fluid. Blood plasma, fluid of the blood, cerebrospinal fluid, fluid surrounding nervous system organs. Extracellular materials also include cellular secretions, such as saliva or mucus, and extracellular matrix, substance that acts as glue to hold cells together. Plasma membrane acts as an active barrier separating intracellular fluid, another name is ICF, from extracellular fluid, ECF. It plays a dynamic role in cellular activity by controlling what enters and what leaves a cell, also known as the cell membrane. So you can see on this diagram, we have a plasma membrane or cell membrane. So remember, cell membrane is not the same as cell wall. Human cells do not have cell wall. It does have plasma membrane. So that's inside the cell. Inside plasma membrane, we have ICF, and cells are surrounded by fluids outside that are called ECF. structure of plasma membrane. It consists of membrane lipids that form a flexible lipid bilayer, specialized membrane proteins. So here's first one, lipids, that create lipid bilayer. Next, membrane proteins that flow through this fluid membrane, resulting in constantly changing patterns, referred to as fluid mosaic pattern. What it means that those proteins inside the membrane, they're not located in one specific spot. They float through this lipid bilayer. On the surface of a cell, the surface covered by sugar called or form glycocalyx. Membrane structure help to hold cell together through cell junctions. I'm trying to get my mouse so I can change the page. Okay, so here you can see a diagram of cell membrane. So this structure with the head and two tails represent phospholipids, and you can see we have two layers, extra or external and internal layer of phospholipids. That's why we call it phospholipid bilayer proteins. So those purple structures are proteins. Then we have this yellow structure. This is cholesterol. And these green structures are sugar. When sugar is attached to protein, we call it glycoprotein. Here's our glycoprotein. When sugar is attached to a lipid, we call it glycolipid. And those glycoproteins and glycolipids made glycocalyx. Okay, so now let's talk about those lipids that make our membrane. So lipid bilayer is made of 75% of phospholipids, which consist of two parts, phosphate head and phosphate heads and fatty acid tails. All right, so phospholipid would look like it has a head and it has two tails. Phosphate heads are polar, they are charged, so they are hydrophilic, water-loving. Fatty acid tails are nonpolar, no charge, and they are hydrophobic, water-hating. 5% of lipids are glycolipids, lipids with sugar group on outer membrane surface, and about 20% cholesterol, that is also a type of lipid, that increases membrane stability. Now, membrane protein they have several functions. They allow cell communication with environment, make up about half the mass of plasma membrane. Most have specialized membrane functions. Some float freely, and some are tethered to intracellular structures. So we have two types of membrane protein, integral proteins and peripheral proteins. Integral proteins firmly inserted into membrane. Most are transmembrane, so that means they go across phospholipid bilayer. They span a membrane. Function as transport proteins, channels and carriers, enzymes, or receptors. Peripheral proteins are loosely attached to integral proteins, include filaments on intracellular surface used for plasma membrane support, function as enzymes, motor proteins for shape change during cell division and muscle contraction, and cell-to-cell connections. So on this diagram, you can see different type of proteins and their functions. So those proteins, like a protein over here, is called transmembrane protein because it goes across this phospholipid bilayer. And it's also integral proteins. And some proteins, peripheral proteins, are not shown over here, but it can be, for example, it can be proteins attached right here or attached to inner surface of the membrane. Those gonna be the peripheral proteins that's shown over here. Let's go to previous one. Right. So this one, for example, over here, or this one over here. Those are peripheral proteins. Okay, so this protein's function is attachment to the cytoskeleton and extracellular matrix. Inside, it's attached to another protein that are made cytoskeleton. Outside, they're attached to extracellular matrix protein. This protein is signaling. So here we have a signal molecule, some messenger that activates this protein, and you can see signal passes inside the cell. This is a transport protein. It's a channel, so molecules can go in or out of the cell. Intercellular joining, that's another function. So we can have two proteins that make a connection and hold two cells together. Cell-to-cell recognition, when we have a protein with a sugar attached, so it's a glycoprotein, and another protein of another cell, for example, white blood cell, gonna recognize the sugar and recognize a cell as a self cell. And over here, we show proteins that are enzymes. So function is enzymatic activity. Glycocalyx consists of sugars, carbohydrates sticking out of cell surface. Some sugar are attached to lipids, and we call them glycolipids. I already told you about this. And some to proteins, glycoproteins. Every cell type has different patterns of this sugar coating, functions as specific biological markers for cell-to-cell recognition, and allows immune system to recognize self versus non-self. And on a diagram here, you can see electron microscope picture of glycocalyx and a diagram of the sugar that attached on the periphery of the cell attached to is a protein or lipids, glycoprotein and glycolipid. Right, so here, that's this is a protein, so this is glycoprotein, and this sugar attached to a lipid, so glycolipid, and together the sugar structure make glycocalyx. Homeostatic imbalance. Glycocalyx of some cancer cells can change so rapidly that the immune system cannot recognize the cell as being damaged. Mutated cell is not destroyed by immune system, so it is able to replicate. Just to add to this, here's the cancer cell, and you do have cells of your immune system, natural killer cells. Their function is to recognize and destroy cancer cell. Now, to do that, to recognize the cancer cell, natural killer cell must, it's shown over here. So there's lots of receptors, and some receptors need to be activated, and some receptors should not connect to this sugar on on a cell. So imagine that's somebody's cell, a person's cell that has lots of glycocalyx, lots of receptor, and this need to be precise boundary between natural killer cell and a cancer cell. So natural killer cell secrete a molecule that will destroy the cancer cell. Now, if you see over here, cancer cell, they change this glycocalyx by attaching this molecule over here, different molecules to their glycocalyx. Now, natural killer cell is confused over here, where we don't need, so for example, over here we need activation and we don't have activation anymore. This we have inhibition when we should not have this inhibition. So when natural killer cells cannot recognize glycocalyx on a cancer cell, it cannot destroy it. Some cells are free, not bound to any other cells, for example, blood cells or sperm cells, but most cells are bound together to form tissues and organs. 3 ways cells can be bound to each other include tight junction, desmosomes, and gap junctions. Tight junction form when integral proteins on adjacent cells fuse to form an impermeable junction that encircles whole cells. Tight junction prevent fluids and most molecules from moving in between cells. So you can see on this diagram, the tight junction shows right here. When we have a membrane of adjacent cells fused together by those integral proteins, so the substances cannot pass between the cells. A tight junction is impermeable junctions that form continuous seals around the cells, prevent molecules from passing through the intercellular space. Desmosomes are rivet-like cell junctions formed when linked proteins of neighboring cells interlock like the teeth of a zipper. Desmosomes allow a give between cells, reducing the possibility of tearing under tension. So desmosome anchoring junctions that bind adjacent cells together act like molecular Velcro and also help form an internal tension-reducing network of fibers. So you can see the desmosomes are shown right here, where we have a special linker proteins that attach to internal proteins inside the cells and join cells together, but giving it a little bit of a give that allow to stretch tissue slightly. Gap junction are transmembrane proteins that form tunnels that allow small molecules to pass from cell to cell, used to spread ions, simple sugar, or other small molecules between cells, allows electrical signals to be passed quickly from one cell to the next cell, and used in cardiac and smooth muscle cells. And not only, but you can find them in a cardiac and smooth muscle cell to pass electrical signals, right? But you also will find it in many other cells of your body, like epithelial cells, for example. Those are, by the way, epithelial cells that show you all 3 types of junction: tight junction, desmosomes, and gap junctions. So here you can see proteins that form channels, allow molecules to move from one cell to another cell. Gap junction, communicating junctions that allow ions and small molecules to pass, are particularly important for communication in heart cells and embryonic cells. Now we covered cell membrane. Let's talk about cytoplasm. Cytoplasm are all cellular material that is located between the plasma membrane and the nucleus. Cytoplasm composed of cytosol, that is gel-like solution made up of water and soluble molecules such as protein, salt, sugar, etc. Inclusions, insoluble molecules, example, glycogen granules, pigments, lipid droplets, vacuoles, crystals, and all the organelles. The organelles are metabolic machinery, machinery structure of cell. So cytoplasm made of cytosol, that is pretty much like a fluid portion of cytoplasm, organelles, so cytosol, organelles, and inclusions. Cytoplasmic organelles include membranous and non-membranous organelles. Membranous mitochondria, endoplasmic reticulum, Golgi Apparatus, Peroxisomes, Lysosomes. Non-membranous, that means there is no phospholipid membrane surrounding organelles, include ribosomes, cytoskeleton, and centrioles. So we're going to start with mitochondria. Mitochondria are called the power plant of cells because they produce most of cells' energy molecules, ATPs, via aerobic oxygen requiring cellular respiration. Mitochondria are enclosed by double membranes. Inner membrane has many folds called cristae. So you can see a diagram and a microscope, electron microscope picture of mitochondria. So the major function is production of ATP. Ribosome are non-membranous organelles that are site of protein synthesis. So function of ribosomes to synthesize proteins. They made up of protein and ribosomal RNA that abbreviated as rRNA. Two switchable form found in cells, free ribosomes, free-floating ribosomes, so they inside cytoplasm, and membrane-bound ribosomes. They attach to membrane of endoplasmic reticulum. So if ribosomes attach to ER, right, those are membrane-bound ribosomes. If they're not attached, if they're just inside cytoplasm, they are free-floating ribosomes. And on the diagram over here, you can see two subunits of ribosome, large and small subunit. Those are proteins, right? So here, and there is also ribosomal RNA inside this organelle. Endoplasmic reticulum consists of series of parallel, interconnected cisterns, flattened membranous tubes that enclose fluid-filled interiors. So if you look at the picture over here, you can see the nucleus. So here's the nucleus. We have nuclear envelope. That's a membrane of the nucleus. And continuous, we have this the system of tubules, system of tubes that called endoplasmic reticulum. Endoplasmic reticulum, or ER, is continuous with outer nuclear membrane. And we have two varieties, rough ER and smooth ER. So you can see this one over here is rough ER, and this one is smooth ER. So let's see the difference between these two. Rough ER. External surface appears rough because it is studded with attached ribosomes. So why we call it rough ER? Because we have these ribosomes attached and it has this rough appearance. Now function is synthesis of proteins that will be secreted from a cell. Well, ribosomes synthesize proteins. A rough ER is a site of synthesis. Right? So when ribosomes synthesize new protein, it's deposited inside the lumen of rough ER. But not all the proteins, but proteins that will be secreted from cell. site of synthesis of many plasma membrane proteins and phospholipids. A smooth ER is network of loop tubes continuous with rough ER. Its function is in lipid metabolism, synthesis of lipids, cholesterol, and steroid-based hormone synthesis. Synthesis of glycogen, absorption, synthesis and transport of fats, detoxification of certain chemicals such as drugs and pesticides, and storage and release of calcium. So that's our smooth ER. You know, I'm sorry, over here, this is not correct, because rough ER, I don't think it synthesizes phospholipids. I need to double check, but I'm pretty sure phospholipids are synthesized inside a smooth ER, because that smooth ER function is a lipid metabolism. I think this is just the typo in the PowerPoint. And another thing, I want to go back for a moment, back to ribosomes. Now, if I have entire cell, I want to go back to the diagram of entire cells. I don't want to see if I can just to see all my PowerPoints. I want to show you, go back over here. And I want to show you on this diagram that ribosomes are actually found in the cytoplasm. Now, what is cytoplasm? Cytoplasm is all this area here, from plasma membrane to the nuclear envelope. So all these organelles are part of cytoplasm. The fluid portion is cytosol. So you can see the ribosomes, those are free-floating ribosomes. Those are attached ribosomes. They are inside cytoplasm, but the ribosomes are assembled inside the nucleus. So inside the nucleus, maybe we will have it. It has the nucleolus. By the way, here, that's a nucleolus, and ribosomes are assembled inside the nucleus, and then they moved out to the cytoplasm. It is possible that I have it in my future slides, but just in case, I want to mention it right now, and then we can repeat it if needed. Okay, so next is Golgi Apparatus. Stacked and flattened membranous cistern sacs. Function of Golgi Apparatus: it modifies, concentrates, and packages proteins and lipids received from rough ER or from smooth ER. If it lipids and it's received from smooth ER. So you can see this shown here like this green structure. It's not green, they just use color green to show it to us. We'll see this vesicle coming from rough ER. And whatever product we have inside this vesicle, it's deposited inside the lumen of Golgi apparatus for processing, modification, and then it packages into secretory vesicles, and this secretory vesicle moving towards cell membrane and brings this newly synthesized product, proteins, for example, to secrete it out of cell. So this diagram shows us how the sequence of events from protein synthesis on the rough ER to the final distribution of those proteins. So we can see that ribosomes attached to rough ER synthesize proteins. These proteins now inside these cisterns, inside this lumen of rough ER, is called cistern. Protein containing vesicles punch off and migrate to fuse with the members of Golgi Apparatus. So here they fuse. Proteins are modified within Golgi compartments, and then protein then packaged within different vesicle type called secretory vesicle, depending on their ultimate destination. So yeah, secretory vesicle, if we secrete, so secretory vesicle fuses with the plasma membrane and secrete products through exocytosis, or it can be vesicles that form lysosome. So we have digestive enzyme inside, and lysosomes can fuse with a phagosome and digest whatever is inside phagosome, for example, bacteria. Also, from the Golgi apparatus, we can have vesicle that fuse with the membrane and incorporate in the membrane. And we need it if the cell is growing, for example, we need to make this membrane longer, or to replace the part of the membrane that might be damaged. Peroxisomes are another organelles. They are membranous sacs containing powerful detoxifying substances that neutralize toxins. There is two main detoxifiers or enzymes: oxidase, that uses oxygen to convert toxins to hydrogen peroxide, H2O2, which is itself toxic. And the second enzyme is called catalase. And catalase converts hydrogen peroxide to harmless water. So on this diagram, you can see cell of a liver. It's a red liver cell. And first you have a normal cell, and red exposed to chemicals, you can see increased number of peroxisomes, because if we have toxin in our body, the liver gonna break down those toxins, hopefully convert them into water, right? And to do it, liver cells need to increase number of peroxisomes in their cytoplasm to detoxify harmful substances. Lysosomes are spherical membranous bags containing digestive enzymes, acid hydrolases, those are enzymes, and plus it might be a different enzymes, the enzymes that break down proteins, sugar. Lysosome digest ingested bacteria, viruses, and toxins, degrade non-functional organelles, remove cellular debris, extracellular release in injured causes cells to digest themselves, autolysis. So when the lysosome birth open inside the cell, so here you can see the cell, the cytoplasm, and lysosome. So this lysosome can actually release their enzyme inside the cytoplasm and destroy entire cell. And this will be called autolysis. Auto means, well, I mean, lysis means breaking down, and autolysis means cell break down itself. Cell destroy itself, autolysis, because of lysosome. So here's some homeostatic imbalances. Lysosomal storage diseases result when one or more lysosomal digestive enzymes are mutated and do not function properly. So remember, lysosomes, they have digestive enzyme to digest and break down different chemicals. When those enzymes are not non-functioning enzyme, if they do not function properly, then this can result in a disease. Example of such a disease is Tay-Sachs disease. It's a condition in which the patient lacks a lysosomal enzyme needed to break down glycolipids in brain cells. Glycolipids build up as a result of this defect. So we don't have enzyme to break it down, so we start accumulating these glycolipids. we build up those glycolipids in brain, and it's interfere with nervous system functioning, causes seizures, mental retardation, blindness, and death before age 5. So you can see a normal brain shown over here and the brain of a patient with Tay-Sachs disease. Cytoskeleton is another organelle. It's non-membranous organelle. It's elaborate network of rods that run throughout cytosol. Hundreds of different kinds of proteins link rods to other cell structures. Also act as a cells, bones, ligament, and muscles. Of course, the cytoskeleton is made of proteins. It's not a bone, ligament, and muscle. That's why we have parentheses over here. But they play important role in the movement and support. There are three types of cytoskeleton, three types of proteins that make cytoskeleton: microfilaments, intermediate filaments, and microtubules. Also part of the cytoskeleton are motor proteins, complexes that function in motility, can help in movement of organelles and other substances around cell, use microtubules as tracks to move their cargo on, and they're powered by ATP. So on this diagram, you can see this is our cytoskeleton. And here's the motor protein that attached to cytoskeleton and carries this vesicle inside a cell. Centrosomes is a microtubule organization center consisting of a granule matrix and centrioles. Centrioles are a pair of barrel-shaped microtubular organelles. Now, inside animal cells, we have centrosomes shown right here, right? Just a second. Right there, and then they kind of magnify this area so you can see that we have this granular matrix, right? That's a granular matrix, and pair 1, 2 barrel-shaped centrioles. And the function of the centrioles is to form the spindle called mitotic spindle. So those centrioles, they will migrate to opposite pole of a cell. So one stay here, one migrate over here, and they will form mitotic spindle that allow a cell to divide. So newly assembled microtubules, that is this, microtubules, radiate from centrosome to rest of cells. Some microtubules aid in cell division, and some form cytoskeletal tract system. Centrioles form the basis of cilia and flagella as well. Now about cilia and flagella. Certain cells have structure extending from a cell surface. That's what cilia and flagella is. It's a structure that you will find on the surface of a cell, like over here or right there. They aid in the movement of the cell or of materials across the surface of the cell. That's a cilia or flagella. Значит, cilia, remember, cilia and flagella are for movement. In a human body, we do not move cells. There is only 2 cells in our body that move. Those are white blood cells and sperm. Sperm have flagellum, and flagellum moves the sperm cell through the duct system to the fallopian tubes for fertilization, right? So flagellum moves a cell, sperm. Cilia does not move a cell in human body. It moves substances or materials across the surface. For example, this mucus. Microvilli are also cellular extensions. They are finger-like projections that extend from the surface of the cell to increase the surface area. And microvilli are not for movement, they are for absorption. So they increase the surface area so we can absorb substances from, for example, lumen of GI tract to the cell, and through the cell, then to our bloodstream. So cellular extension, cilia and flagella for movement, and microvilli for absorption. Now, nucleus. Should have started with the nucleus, but now when we know the rest of the organelles, let's focus on one of the largest organelle inside eukaryotic cell. Nucleus is the largest organelle, contains the genetic library of blueprints for synthesis of nearly all cellular proteins. Responds to signals that dictate the kinds and amounts of proteins that need to be synthesized. So most of the DNA of eukaryotic cell located inside a nucleus. Most of our cells are uninucleate. That means they have one nucleus per cell. But skeletal muscle, certain bone cells, and some liver cells are multinucleate. They have many nuclei, several nuclei, or many inside one cell. Red blood cells are anucleated. They do not have nucleus. The nucleus has three main structures: nuclear envelope, that is the double membrane covering nucleus, nuclear membrane or nuclear envelope, nucleoli, so that's a nucleolus. Now, nucleolus is singular, nucleoli are plural. So some cells have several nucleoli and some have one nucleolus, and chromatin, and chromatin is the DNA. Nuclear envelope is a double membrane barrier that encloses the jelly-like fluid, the nucleoplasm. Nuclear pores allow substances to pass into and out of nucleus. Those structures shown here, those oval or round structures, are nuclear pores, allow substances to move in and out of nucleus. Nucleoli are dark staining spherical bodies within nucleus that are involved in ribosomal synthesis and ribosomal subunit assembly. Remember, I told you that ribosomes are found in the cytoplasm, but they are assembled and ribosomal RNA are synthesized inside nucleolus, so inside the nucleus. So usually, we already mentioned it before, one nucleolus or two nucleoli per cell. Chromatin. Chromatin is found inside nucleus. It consists of 30% thread-like strands of DNA, 60% histone proteins, and 10% RNA. So chromatin is a structure made of DNA, proteins, and small amount of RNA. Chromatin arranged in fundamental units called nucleosomes, which consist of DNA wrapped around histones. Chromosomes are condensed chromatin. Its condensed state helps protect fragile chromatin threads during cell division. So you can see over here the genetic material is our helix, double helix DNA. So here's the DNA molecule. Then we have histones, and DNA molecule is wrapped around the histones, making a structure called nucleosomes. And this entire structure made of DNA, histones, and small amount of RNA is chromatin. Now, this is how chromatin look like inside a nucleus when cell is not dividing. It's in the form of chromatin. So it's like the ball of spaghetti, and you cannot really see where individual chromosomes are. But when cell is ready to divide, this chromatin will condense and assume a form of chromosome. So chromosome is just condensed chromatin in a cell that is ready to divide. So I have 3 more slides to go. Autophagy. Cells that have become obsolete or damaged need to be taken out of system. So if your cells cannot function properly, if they are damaged, you do not want the cells inside your body. There are several processes how to get rid of non-functioning cells. One of them is autophagy, that means self-eating. Is the process of disposing of non-functional organelles and cytoplasmic bits by forming autophagosomes, which can then be degraded by lysosome. So on the diagram, you can see how we form autophagosome from the endoplasmic reticulum. The membrane kind of surround the organelle that is not functioning. Then there is a fusion with the lysosome. Lysosome secrete digestive enzyme that break down this non-functional organelle. Autophagy, self-eating. Another way to get rid of cells that you don't need is called apoptosis, also known as programmed cell death, that causes certain cells, for example, cancer cells, infected cells, old cells, to neatly self-destruct. Process begins with mitochondrial membranes leaking chemicals that activate enzymes called caspases. So apoptosis does not involve lysosome. Autophagy rely on lysosome, right? Apoptosis begins with, what do you think? With mitochondria, right? So the organelle that important in apoptosis is mitochondria. When enzymes or chemicals leaked out of mitochondria, enzymes called caspases, they cause degradation. The chemical leaks from mitochondria and they activate caspases. The caspases cause degradation of DNA and cytoskeleton, which leads to cell death. Dead cells shrink and is phagocytized by macrophages. So this is another way to get rid of cancerous, infected, or old cells. So on this diagram, you can see 2 processes, apoptosis and necrosis. In the apoptosis, the cell is actually shrinks, right? And no inflammation. It's blebbing. That means it's broken apart. It's became smaller, and you can see some parts break off the cell. So the cell breaks apart into several apoptotic bodies, which are then phagocytized, no inflammation. When necrosis is death of a cell, but it's not controlled, it's not programmed. Cell swells and plasma membrane rupture causes inflammation. So that's a difference between apoptosis and necrosis. And that was the last slide that we covered. Okay, so I just wanna remind you that we cover cells, the living units, And thank you for watching. I hope it was helpful.